So you have made it to your official final biology lesson. This is section 1.4, part 2. So in the last set of notes, we talked about making GMOs. So now we're going to look at why you would want to create GMOs, what are some benefits, and what are some of the risks associated with GMOs. Okay, so uses for GMOs. Since the 1980s, the technology has been used to, number one, the technology has been used to make copies of genes or proteins. In 1982, Genentech modified bacteria to produce insulin for human use. Insulin is used to manage diabetes by reducing high blood sugar levels. So normally the human body produces insulin, but if you're diabetic, your pancreas doesn't do a good job of creating insulin to regulate blood sugar. So as a result, you inject insulin to correct this. So instead of this insulin being made by your pancreas, it's instead made by bacteria that have a gene inserted into them for insulin production. This was the first widespread use of GMOs. A human cell was used, and they were able to isolate the insulin gene, then take a plasmid, then insert the insulin gene into that plasmid, which then gives us a transgenic bacterium, which you could grow on a plate in culture to produce more transgenic bacteria. And then from that, you can extract the insulin which is produced, which can then be used for pharmaceutical use. So that's just what the bottom image there is showing. Another use of GMOs is to determine gene function. So this is basically what are genes used for. So they do this through what is called a knockout mouse, which is where they break the gene of interest in order to study the function of the gene. So if the gene is broken, meaning it's not expressing what the gene should be expressing, it can't perform its function. So then you can see what that function is lacking or has lost. So we take our wild type mouse, which is an example of a normal mouse without any changes made, and then you figure out your gene of interest for a particular function, for example, fur color or number of toes. Then you knock that gene out, so meaning we stop it from working. So when that gene is no longer expressed properly, let's say for creation of fur, the knockout mouse wouldn't have any fur when you compare it back to the wild type mouse, which is the normal one. So by looking at the knockout, you can determine what a particular gene of interest does. So a third potential use for GMOs is by studying gene expression patterns by modifying genes to see how the expression changes through the entirety of an organism's life or in different organs or regions of the organism. So for example, mutating a plant gene, ATPLC1, so that it is amplified and then measuring its expression and effect during the many life stages of the plant. So making a plant gene so that it's amplified, meaning that there's more of it, it's more prominent, and then measuring its expression effects during many stages of that plant's life cycle. So being able to see as the plant progresses how much the gene is actually being amplified at these different stages. So compare this, for example, to human growth hormone, in which it is much more important for a baby or a child because it allows them to grow bigger than this gene for growth hormone would be at an adult stage where we've stopped growing. So we see that the gene is being expressed more as a child or baby than as an adult in a sort of tapering off pattern. So this use of GMOs basically allows us to get more information about different organisms and when different genes are actually being expressed, because some will be turned on or off with age. Our fourth function is to create models for human traits and genes. So for example, modifying mouse genes that are identical or similar to human genes to determine what the genes do. So because we are unable to obviously create a knockout human, we are able to modify mouse genes to figure out what the effect would be on a human. So basically, what does a particular code do in a human? So let's take this code, insert it into the mouse, and figure out what it does so we can report what it would do in a human, essentially, to put it in very simple terms. Our last common use is to generate modified food sources, both plants and animals. So this is probably the most common thing we think of when we think of GMOs. So for example, Bt-resistant corn. So Bt-resistant corn is a genetically engineered plant that produces insect toxin called cry protein. Crystal-like proteins is what that stands for. The Bt gene comes from a bacteria called Basilicus thuringiensis and produces a toxin that disrupts gut function in the larvae, so essentially the caterpillars, of certain insects that are crop pests. So there are dozens of insects considered to be crop pests. There is a bacteria that normally produces a toxin that would make it so the larval stages, i.e. caterpillars, can't digest food properly. So basically, they have taken this toxin that the larvae can't digest and put it into the leaves of the corn so that those larvae can't eat the plant. And basically, they can't eat it all. So you isolate your gene out of the bacterium, insert it into your crop, and then the pests will die when they eat any plant parts. So you end up with the insect not being able to destroy the plant. So that's what that bottom image is showing. Another example is golden rice. And it is called golden because you can see it's visibly much more yellow than standard white rice. So in 2000, researchers in Switzerland developed a genetically modified strain of rice known as golden rice. Golden rice has an increased iron and vitamin A content, and it is designed to help reduce malnutrition in poorer parts of the world. So the idea was that rice is easy to grow. If you can get more nutrients from rice, then you can have people being fed nutrients easier. So golden rice has 4 different foreign genes. It has its own chromosome. It has something called ferritin, which increases iron content. It has an enzyme that makes it easier to absorb iron, and then a protein that makes it easier to increase your iron uptake. So you're putting iron into your body, you're getting rid of something that makes it hard to absorb iron, and you're adding something that makes it easier to absorb iron. And then finally, you have genes for beta carotene, which is a precursor for vitamin A, to increase your vitamin A intake as well. So there are lots of different genetic traits that have been expressed in the United States. Apples, for example, have been made so that they don't brown as easily. So this is referring to how when apples are cut and exposed to air, they turn brown pretty quickly. So they have been genetically modified to prevent this from happening. Not that there's anything wrong with eating apples that are brown or have turned brown after being kind of oxidized, but it just doesn't look very appealing or appetizing. Potatoes, to reduce bruising and black spots, once again, non-browning. Blight resistance, meaning making it much more likely to have a good crop. Corn has been made with insect resistance, herbicide tolerance, meaning you can kill the weeds, but you can keep your crop alive. There are also drought tolerance genes to prevent the plant from dying if it gets really dry. Canola has herbicide tolerance, again allowing it to be sprayed without killing it off. Alfalfa has herbicide tolerance, soybean insect resistance, herbicide tolerance. You can see a trend here. Papaya has disease resistance, and without that, it wouldn't be able to survive. Usually, this is just ways to make it so that the crop will either thrive, or so it's easier to maintain the crop, or to make it so that the product will last better. So that's kind of the purpose of this genetic modification. Okay, so another example is transgenic goats that make human growth hormone in their milk. Goat's milk is another source of calcium and protein for humans, so that's why it was used for this purpose. So goats have been genetically modified to make human growth hormone in their milk. This hormone is used to treat humans who have a non-functioning gene and without treatment would have shorter stature. And this is referring to significantly shorter than average. So more than 2 standard deviations below the average. So this isn't just a little bit short, this is quite a bit shorter, not just because like your parents are short, for example. So basically, if a human has a non-functioning gene for a human growth hormone, and that has been identified as having a deficit, they can have this milk in their diet as a supplement in this goat milk that has human growth hormone in it to kind of help them grow. Or another example, transgenic pigs that are more compatible with human tissues and can be used as organ donors. So basically making it so that you can have pigs grow human organs or very close to human organs that can be used as a transplant. So this is giving an example of the human pancreas in this image being created in a pig so that it can be donated to a human. The pancreas is an example of something that humans only have one of, and it is needed for survival. So it can't be a live donation from human to human. Also, the pancreas doesn't survive well post-mortem, so after death. So this allows for potentially more options for human organ donations, not necessarily coming from humans. Okay, so below are some advantages and disadvantages of GMOs. What I would like you to do is pause, and there are 2 slides for this, so just be aware of that. And put a check beside the ones you think are advantageous and an X beside the ones you think are disadvantageous. So some of these may be seen as more advantageous to some people and less to others. And this doesn't necessarily mean that it's right or wrong. But some of these will have more obvious answers than others. So it's just looking at the ones that you think are potentially a good thing and ones you think are potentially a bad thing. So pause here if you need to. This is also in your notes. So again, this is from page 81 in your notes. So feel free to pause here and discuss with others and ask questions if you need. So when we're talking about GMOs, we're usually talking about the ones done through technology of cutting out and stitching together genes. But genetically modifying food has actually been happening since the beginning of agriculture, so over 12,000 years ago. But it was previously done through what is called selective breeding. So we breed our plants and animals in order to make them more likely to have traits that we want, more desirable traits. Modern technology speeds up this process, and most scientists agree that GMOs and GMO foods are safe. But this doesn't mean that we should genetically modify absolutely everything. So, however, this does not mean the technology should be given a blanket approval. GM crops have been planted extensively for little over a decade. While no negative health consequences have been detected or are anticipated, it is the relative newness of GM crops requires that we need to continue to monitor for health impacts. Because this is so brand new, we don't know what the future holds. Science is constantly changing. From what we know right now, GM crops are completely safe, but we don't want to start messing around with too many things and allowing everything to be genetically modified because we don't know the potential future impacts, good or bad, that they could have. So it's just making sure that we're doing this in a controlled way. So there are some assigned readings on artificial versus natural selection as well in your notes that I would like you to do. But this is the end of section 1.1, part 4.